Oh. Wow. I feel like sort of rock star with this. So thanks for coming. So I'm going to talk today actually about octopuses. For those of you who have seen any of my talks before, I've been primarily focused on voltage-gated sodium channels in salamanders, but we branched out in the last year or so, and I think we're seeing some kind of interesting things here related to convergence and parallelism associated with TTX and TTX resistance in these guys. Briefly, for those people who aren't familiar with it, the molecule that I've been interested in for a decade or so now is this molecule Tetrodotoxin. This is a small molecular weight toxin that blocks the initiation and propagation of action potentials in nerve and muscle. It's present in a huge array of taxa, marine taxa like blue-ring octopuses, pufferfishes. It's also present in salamanders and some frogs. One of the sort of deep questions about this is what's making it, where it's coming from. I'm not going to touch on that today, but it's kind of a weird toxin. This thing targets voltage-gated sodium channels. In blue-ring octopuses, these guys have moderate to high levels of TTX. In these guys, they're both poisonous and venomous. They have it in ink, they've got it in their eggs, they've got it in their mantle, but they've also got it in their post-salivary glands, which they're likely using to inject into prey. So these guys, and I emphasize that here because just like newts and just like pufferfish, these guys use TTX as this sort of central component of their ecology. They're not like snakes that sort of opportunistically eat newts, right? It's this central thing. And I think that that's relevant in terms of understanding this question about resistance, right? They have it, it's integrated into their system, so they have to be resistant to it. And with all of the vertebrates that we've looked at, right, resistance comes from evolution, adaptive evolution of TTX-resistant voltage-gated sodium channels. So, as I said, these voltage-gated sodium channels, these are ubiquitous in animals. They govern the propagation and initiation of action potentials in nerve and muscles. These proteins are composed of 4 homologous domains with a central pore that sodium passes through, and as the positive sodium ions pass through, it generates bioelectrical changes in bioelectrical potential in membranes. What I've highlighted here are these pore domains. This is a molecular model of the channel, and TTX binds in this pore region and occludes the pore and prevents sodium from moving through. And so we know that there are specific interactions in some positively charged amino acids in here that can disrupt the ability of TTX to bind. So we have a really nice sort of zone to look to understand what's going on here. And in fact, in salamanders that have TTX, we know that there are a subset of 4 mutations in domain 3 and domain 4 of the pore that make these things super resistant to TTX. So here you see data here. This is 300 micromolar TTX. As a reference, 10 nanomolar TTX is enough to completely block action potentials in a frog, right? And that really comes from these 3 substitutions. So we were interested in 2 questions here, right? First, establishing that blue-ring octopuses are actually resistant to TTX. And second, right, does resistance derive from adaptive substitutions in the relevant voltage-gated sodium channel? So we used electrophysiology. We did whole cell patch clamping on dissociated neurons from the stellate ganglion of blue-ring octopuses. And then we used an outgroup. This is a California species that theoretically is non-toxic. And then we generated transcriptomes from the optic lobe and stellate ganglia of a blue-ring octopus and then a Pacific brown octopus that co-occurs with these guys in Bali and does not have TTX. So what did we find? Well, here what you can see is sodium currents in response to 100 nanomolar concentrations of TTX. And in black, right, these are not exposed to TTX, and then blue in the octopus is the TTX, and then red in the blue rings is exposed to TTX. And what you can see, I think very clearly, is that this concentration of TTX had no effect on the sodium currents in the blue ring octopuses. And yet in octopus and rubescens, it reduced it to about 25%. Right? So again, if this were a frog, 10 nanomolar TTX would have completely flatlined it. So we're seeing this kind of weird thing that we don't understand yet in octopus, that it's sort of low-level resistance We looked at this in a little higher concentration, so this is 10 micromolar TTX, and again, no effect. So I would argue that these guys fall in this category with newts and pufferfish of basically these super resistant organisms. Not surprising given that they have TTX and they use it in their ecology. So that led to this sort of primary question, which was, what about the sodium channels, right? Has resistance evolved through adaptive substitutions in the voltage-gated sodium channel of octopuses, right? And are these changes convergent with those that we see in vertebrates? Something just to introduce you to this, right? In invertebrate cephalopods, like all invertebrates, actually have two NAV channel genes, 1 and 2. And NAV1 is homologous to the whole family of channels that are present in invertebrates. So this is NAV 1.1 through NAV 1.9. These are typically TTX sensitive invertebrates, but there's some differences there. And then NAV2 is actually absent in vertebrates. The reason that I'm emphasizing this is that NAV2 is actually resistant to TTX in all invertebrates, and but it's also permeable to calcium. And that permeability to calcium, as well as TTX resistance, is associated with a single amino acid in domain 3 of the pore. These are of interest to us for a couple of different reasons, but for those of us who are interested in the evolution of voltage-gated sodium channels, we're now thinking that these particular NAV2 is actually an evolutionary intermediate between voltage-gated sodium channels, which is thought to be the ancestral gene family, and the derived voltage-gated sodium channels, right? So what did we find? So we actually identified two channels in blue rings and Pacific brown octopuses. These channels were present in both the optic lobe and stellate ganglion, and they had very different pore structure that was consistent with the presence of NAV1 and NAV2. And when we put them into a gene tree, we had something that was very nice and very satisfying. Down here you can see this cluster here that are the octopus NAV2s, or what we thought were the NAV2s, and they cluster very nicely with a squid channel that's also NAV2, and then they cluster with a series of other invertebrate NAV2s, and then up here you've got the NAV1 channels from octopus clustered with the squid, the other NAV1s from other invertebrates, and then they find a nice sister clade to the rat channels that I put into this tree. So these are all of the rat NAV 1.1, 1.2, and you can see that those are sisters. So these NAV1s are more similar to these vertebrate channels, which is typical for a gene family. So when we looked at the sequence of NAV2, what we found was, in fact, that there's really very little sequence difference between our Pacific brown here and our blue ring channel. You can see that for domain 1, 2, and 4, these are identical. There's a minor change at this position in domain 3. We still don't understand what's going on here, but this E here in domain 3 is consistent with a TTX resistant channel for NAV2. So one of the questions that we wanted to look at was there was a possibility that in these blue rings that what they were doing was a sort of classic Evo-Devo answer to this, where they're upregulating this channel, right? They have this tool already, but what we found when we looked at the transcriptome was we didn't see any evidence of upregulation of this channel at all. So it made us want to look at NAV1. And that's where I think where we found the money here. And what we find here is, what I want you to see here is, here in blue are TTX-sensitive invertebrate channels. These are TTX-sensitive vertebrate channels, 1.4, 1.7, 1.6. Here's our Pacific brown octopus, and then these are TTX-resistant invertebrates, blue ring, and then for comparison's sake, I have the Tetrodotoxin channels in here. And what we identified were substitutions in domain 3 and in domain 4 that likely underlie the elevated TTX resistance that we see in blue ring octopuses. And so what I want to highlight here is that in inverts, domain 3, there's this isoleucine here. In vertebrates, it's a methionine. In octopus, it's a methionine. In these two TTX-resistant invertebrates, where other people have looked at the TTX sensitivity of these currents, there's actually a threonine here. And in blue rings, there's a threonine. And then in Tetrodotoxin, there's also this equivalent threonine. Same amino acid change, same position. And then in domain 4, in the resistant channels, there's this consistent motif of this aspartic acid and glycines over and over and over and over. And then you get to the psittophragm and it's still there. And then boom, you start looking at these resistant invertebrates, and what you can see is that there are changes here that are conserved within invertebrates, but also conserved across vertebrates and invertebrates as well. And just to summarize that, I have this as a question mark. What I want to show you here before I finish is that here we have TTX-resistant snakes. Here we've got TTX-resistant pufferfish channels. Here I've got nuke data. And down here I've got these resistant vertebrates. And what we see over and over and over again is that this same methionine to threonine substitution. This has occurred at least in 7 different lineages, including blue rings. And then, again, these similar changes. These substitutions associated either with the aspartic acid or that glycine in domain 4 occur in at least 10 lineages. So we're seeing this over and over and over again. And the reason that I have this in terms of convergence here is I would actually argue that this is an example of parallel evolution. Right? Octopuses are significantly divergent from vertebrates, but this NAV1 channel is the homologous channel to that whole gene family. So I think we're seeing something here where there's something about this ancient NAV1 channel and then all of the paralogs in vertebrates where we're seeing some sort of combination of constraint or epistasis that are driving these changes over and over and over again. And that for us is where we're moving in my lab, is we're starting to make the actual changes in these channels and starting to look at the functional effects of these individual changes. So I'm going to end there and take any questions.